Okay, okay. Thank you so much for letting me present my work. I'm a PhD student at Queensland University of Technology in Australia. And today I will talk about DNA aptamer-based biosensor for salivary detection in heart failure biomarker. So for this one, I'll give you a bit of introduction of what is heart failure. So basically, as it's a condition or it's a failure of the heart to pump enough blood around the body, and it's affecting around 30 million of people over the world. And one of the critical things about this condition is that more of the 50% of the patients, they need to be re-hospitalized. So that is creating kind of the burden in the health care system. So despite all the treatments, we have some, of course, common symptoms that we can lead to diagnosis, but they are short of the breath. It could be also fatigue, for example, confusion or memory loss. So as you might notice, these are non-specific symptoms, and they are, of course, leading usually in late diagnosis, and they are impacting in the healthcare. So these days, we are monitoring and we are diagnosing through more or less an electrocardiography and electrocardiographic. However, some biomarkers, especially the peptides, have been studied in some immunoassays, but for this, we still need blood collection or blood sample. So there is, of course, a clinical gap because we are taking blood from the patients and we are limited in the points of need. And of course, we have some delay in the diagnosis. So when we think on this gap, we also can bring the idea of use saliva as a potential sample and discovery biomarker for heart failure. So saliva shares more than 25 of the contents of the biomolecular content with blood, so makes this a valuable medium for an alternative also. And it's been studied a few biomolecular biomarkers. Some of, for example, the NT-proBNP, and also it's being galectin-3, S100. They are proteins that we are going to focus on. Our group has been working with S100A7 and also galectin-3. So I will start with S100, that the protein has been reported and demonstrated to be twice the concentration in patients that they are experiencing heart failure compared with individuals like they are healthy. So in our group, we are doing the selection from scratch. So because S100 is a protein, we use the traditional selects. So we use a library that contains a random version of 40 nucleotides. And we perform a negative selection just to get rid of unspecific binding to the streptavidin on the magnetic beads. Then we perform the positive selection, that in this step, we are adding the target of interest in. And we perform the enrichment through 10 rounds of selection. If you go through the literature, some people are doing between 8 or 10. We went for 10 rounds. And then the pool was sent into NGS, and we analyzed the data that we got. And it was one aptamer that we call it aptamer 17, which took over the 50% of the pool. So it was the highest one. And a bit of a spoiler, it was kind of the best kind because we did some stuff with the aptamer. And we try, we have available ELISA, ITC, and BLA to perform in the lab. And despite of the difference in the absolute number, we got a KV in the nanomolar ratio. So we went for the idea of one of the great things that we can do with aptamers that we cannot do with antibodies, and we tend to compare them. It's do some modification post-select. So in this stage, we saw what we can do to improve the affinity, if there is something, and of course, always there is something in scientists. So we went to analyze NGS data, and we saw that the modification that we could perform were close to the 3N terminals because we got high variations there. So we perform a bunch of truncations, and the best one is called 1782 because that is the full length of the aptamer. And when we cut it to the 43 nucleotides, the KD improved from 80 nanomolar to 27 nanomolar. So this showed that that version was not important and even better without the version because improved the affinity. But S107A is just is a protein that is a dimer. So we thought we can actually create a dimer of this aptamer and we can have a better affinity. So for this one, we use a poly T and or poly A, both of them linkers. And in the protein, the calcium binding loops is around 28 Armstrong. That is roughly a DNA-based pairs. So we try different lengths. We try 5, 10, 12, and 14 nucleotides. And we got that doesn't matter if we use T or A, but the length needs to be 10. And for example, with the shorters, that is 5, that abolish completely the binding. And with this one, we got a KD of 5.5 nanomolar. This is a prediction that we got it using AlphaFold. It's a lot of contradiction when we use AlphaFold, but with the aptamer and the protein interaction, it suggests that the aptamer is interacting with the N terminal of the protein, and probably these positions are critical. So we went and we tested this one. We did truncation here on mutagenesis and abolish the binding. I'm not gonna go really deep in this one because also we hypothesized and we proved that the secondary structure is G quadruplex. So we have published this one and the size that we did it to get through. And I'm gonna move forward a little bit because the idea is to develop a sensor, right? So we perform first an aptamer or only aptamer ELISA sensor in which we use as a capture and as a detection element the aptamer. And because we wanted to test in saliva, we spiked 5% of saliva, and we got a KD in the 50 and 60 nanomolar ratio. And the limit of detection is around, let's say, 11 nanogram per ml. It was a study that was published, not from our group, that they used an antibody for S100SA ELISA, and they got a limit of detection of 12 nanograms per ml. So this assay is kind of the same or a little bit better, but it still is not enough or doesn't have the great sensitivity for if we're gonna detect in the earliest stage of the heart failure condition. So also a disclaimer here, we only use the full length and the aptamer short. We didn't use a dimer because we need to have here a system with 2 labels, and that is gonna cost some money, and we have to put that in consideration. So we went only for this one. So we thought, what if we try to make an hybrid assay, and we use as capture the aptamer, but as detection system, an antibody for S100. So indeed, we did that. We use this assay, and we test all of them here. We didn't have a great sensibility with the dimer aptamer, but we got a sensibility really good for the aptamer 43. And we got a limit of detection in the picomolar ratio, so that is great. And we have also high reproducibility compared with the commercial ELISA kit. So I'm running a lot. We wanted to test also if this sensor, the hybrid ELISA sensor, can be performed a long time. It has storage. So we just cover the plates. Some of them we cover with the aptamer capture, some of them we did not. And then we wait just for 2 months and a half, either room temperature or 4 degrees. And we got that pretty much it was no loss of sensitivity of limited of detection. And if we pre-cut the plate, we are saving an hour and a half of work. So the totally work ended up to be 3 hours compared with the commercial kit that took us around 4 hours. So another thing that we did for this essay is because S100A7 is a part of a family of protein that is, for example, we have S105, A, and 9, and they are involved in different processes like proliferation or calcium events. We test how specificity is our aptamer for S100A7. So first, we did ITC for all of them. For S100A9, we didn't have any detection or interaction, so that was great. And we have a weekly binding with AA. So we moved to the ELISA assay, and we didn't have no cross-reactivity with all of them in both of the assay, in the aptamer or sandwich assay and in the hybrid ELISA. Because all of that, the aptamer was working well, and that is a hard thing to have sometimes in the lab. We also performed the BLA that is available in our facility. First of all, the first graphic is just the 3 of the aptamers, full length, the 43, and the dimer, and just in buffer. We play a little bit with the buffer, but contains twin and BSA in a low percentage. And then because we wanted to have a real metric, the main idea is using saliva, we spiked 2.5 of saliva, and we did fraction of concentration from 0 to 10 nanomolar. And the KD slightly went a little bit high, but still we have sensitivity, and it's okay to use it there. And lately, we wanted to use for real samples. So it was a bit hard, all the controls to get samples, but we managed. We didn't get any cell, only 3, but we got some patients with heart failures that they were agreed to the process and gave us some samples. And we got an excellent correlation between the BLA that we perform in the lab, that only takes us 20 minutes to do it, with the ELISA kit. So all of this work has been recently published in February, so it's kind of the new thing that we published. But as I mentioned before, S100 is not the only heart failure biomarker. So we just started like a month ago with galectin-3. Galectin-3 is a monomer and it's around 20 kilodalton. So it was a big challenge for me during the selection. I performed 10 rounds again, and I got from the NGS data 2 kind of candidates that I call it C6 and D2. And I only performed the ELISA, and I got also a nanomolar KD. But again, we've been talking during these days, especially in the aptamer conference, there is so many studies that we need to go through to finally confirm that we have a binder or real aptamer. So this is just work in progress, and hopefully I can get something, or probably just something that I'm gonna put it on the bin in a couple of months. I don't know yet. For conclusion and future work, it's just I really summarize the idea of the first aptamer that we find for S100A7 and the potential application in a hybrid ELISA or in a BLA for real quantification. And future work, we just started with another biomarker that is galectin-3. And I think I did it in that time. I would like to acknowledge to my supervisor, Dr. Elena Iremeva. She was presenting in the aptamer conference, and also Kirill Alexandrov. That's it.